Hi, this is a practical review for the Amylase Lab being held at McKinney campus in spring 2020. Chapters 1 through 6 will come from the lab manual that you've been using in class. Chapter 7 will be coming from the lab bench materials that you're doing online. If you have questions from any of these materials we cover, please contact your lab instructor with specific questions. In Unit 1, you should learn how to do conversions within the metric system. We will not provide this chart for you on the practical. Instead, you should be comfortable using it yourself. Make sure that you understand the three basic base units that we have that we use. Those include the meter for length, the liter for volume, and grams for mass. In this, you should also understand how the prefixes work. So mega, we use a capital letter M, and it stands for 10 to the 6th power. Make sure you can convert between metric units. We have a sample problem down here at the bottom. Convert 45 milliliters to microliters. To do this, you need to figure out where you're starting and where you're going. So I'm starting at milli and I'm moving to micro. You then count the decimal points that you're moving. Here I'm starting, and now I'm going to move 1, 2, 3 decimal points to the right. You then do the same thing in your original question. So 45 milliliters was what I started off. We then move that decimal point 1, 2, 3 spaces to the right. And so this answer becomes 45,000 microliters. Make sure that you can convert between English to metric and metric to English, Fahrenheit to Celsius and Celsius to Fahrenheit using those equations. If we give you a conversion, you should also be able to convert between English and metric. For instance, 60 inches equals how many centimeters? Start off with what you know, 60 inches. You then multiply it by a conversion factor, trying to use the units in such a way so that like units will cancel out and you'll be left with just the units you want. In this case, 1 inch equals 2.54 centimeters. So 60 times 2.54 would give you the answer in this particular equation. Make sure that you can write numbers in scientific notation. Scientific notation always fits the form one number in the ones column, decimal point, everything else, times 10 to some power. For example, if we do these two examples, write the number 567,809 in scientific notation, we would rewrite this as 5 decimal, everything else, 67809, times 10 to some power. We then look at how much the decimal point moved from our original number to the scientific notation number, and it moved 1, 2, 3, 4, 5 places, and so this becomes 10 to the 5th. We can also expand numbers. 4.0008 times 10 to the 5th becomes this. We can see that our decimal point moved 5 places. Next, you should be familiar with lab equipment. We have our stir bars, our beakers, our weigh boats, graduated cylinders, Erlenmeyer flasks, serological pipettes, micropipettes, rulers and meter sticks, micropipette tips, our digital balance, and then our hot plate, the stir plate. Please make sure that you're comfortable with all equipment that we've used in all labs. Accuracy and precision, they're not the same terms, although we do like it when things are both accurate and precise in science. Accuracy talks about actually getting the right answer, right? So accuracy means if the answer is 6, you also got the answer 6. Precision just means that you're consistent. So you're really precise if you get the answer 6, 6, 6, 6, 6, right? You just keep getting the answer 6 over and over again. That's very precise. However, it's not very accurate. Here we have an example of somebody who's very precise. They keep hitting the target at the same spot, but they're not very accurate. Whereas in this last case, we have somebody who's both accurate and precise, where they're hitting the right answer and they're doing that over and over again. Make sure you know how to use rulers to measure. Just as a reminder, sometimes rulers get funny in the way that they're labeled. Those big numbers, 1 and 2, those are actually centimeters. So this gray line, if it were to scale, would be 2 centimeters long or 20 millimeters long. Just as a rule of thumb, the width of your pinky is usually about 1 centimeter, or if you've got inches, an inch is about 2.5 centimeters. As we were measuring, the first thing we did was we calculated the volume of various items. You should be able to calculate the volume of any rectangular solid by taking the length times the width times the height. That will give you a volume in cubic centimeters. Cubic centimeters or centimeters cubed is the same thing as saying cc's, and for water that's also equal to 1 milliliter. We then calculated density. You should be able to do that. That's where we take the mass of an object divided by the volume of the object. From here, we started looking at accuracy. Not all glassware items are very accurate. Most of them will list on their surface how accurate they are. So here we've got this beaker, and you can see that its accuracy is only plus or minus 5%. As we look at measuring up liquid, we want to be aware of the meniscus. The meniscus is this short little indentation that happens in the surface of the liquid because of water tension. As we look at this particular item, let's imagine that it's in milliliters. Here we've got 35, 36, 37, 38, 39, 40 milliliters. The amount of water in this particular example would actually be 36.5 milliliters because we've got 36 and then a half. You read the liquid from the bottom of the meniscus. Make sure that you know how to use the serological pipettes, that you can identify which size of micropipette that you're working with as needed. Next, you should be able to read micropipettes. Each micropipette at the top usually tells you its volume that it's good for. So this one is good from 200 to 1,000 microliters. And we call the pipettes by the largest volume that they can use. This one is called a P1000 because it can hold up to 1,000 microliters. Each micropipette is different. At the McKinney campus, the P1000 is going to have a red number at top in the thousands column, followed by the the hundreds column and the tens column. The P200 will have 3 black numbers, and the P20 will have 2 black numbers followed by a red number. Please make sure as you read the micropipette, you're always reading down. With the micropipette, here we give you 3 examples. They're all set to the same thing, 0 to 1, but this might change their volumes depending on which micropipette you're using. So if we're using the P200 and it's set 0 to 1, that actually means 2.1 microliters. If you're using the P200 where you start with your hundreds column, then your tens, then your ones column, this means you're using 221 microliters. If, however, you're using your P1000, that top value, that 0 that's red, that's your thousandths column, and then you've got your hundreds and tens column. So this means you've got 210 microliters. Absolutely feel comfortable using micropipettes when you get to the practical. From there, we ran an experiment where we looked at the relationship between water volume and mass. You should have started recognizing that there was a direct one-to-one relationship between them. What happened was if something was 340 microliters of volume, then it would weigh 340 milligrams. Or another way of saying that is if you had 1,000 microliters of water, it would weigh 1,000 milligrams. One way to write that is to say that for water, 1 milliliter of water will always weigh 1 gram. Or if we think back to what we learned about volume, 1 gram equals 1 milliliter of water, and that will also equal 1 cubic centimeter. And you should be comfortable with that relationship. Make sure you can analyze data from any of the charts we developed on any of the days in lab. Independent variables are always going to be on the x-axis. Dependent variables are always going to be on the y-axis. You should be able to identify the independent and dependent variables in any experiment. The independent variable is the one that I, the scientist, manipulated. The dependent variable is the one that was the outcome of the experiment. Calculations. You should be able to identify the range and the average of any group of numbers. So for the range, you take the largest number, in this case 100, minus the smallest number, 101, which would give you a range of 99. To get the average, you add all of the numbers up. So 100 plus 10 is 110 divided by 5. So pull out my calculator here. 110 divided by 5 equals 22. The average of this group of numbers is 22. Another word that we use sometimes to describe the average is to say the mean. That just means average. Unit 2, microscopy. Please make sure that you know all of the structures on the microscope and their functions. You should also know how to use a microscope, which knobs to turn at which point, and which lenses to use at which time. Total magnification on a microscope can be calculated by multiplying the ocular lens times the objective. So for instance, if you are using the 10x objective, You would multiply this times the ocular lens, which is also 10x, and your total magnification of that 10x objective would actually be an object that was 100 times larger than it's normally seen. As magnification increases, resolution will increase, depth of field will decrease, and field of view will also decrease. Contrast is a measurement of the differences between light and dark in a sample. We often add dyes to increase contrast. The diaphragm can also adjust contrast for us. The working distance is the distance between those objective lenses and the stage. You have to be very careful at the higher power objectives that you are aware of that working distance. On page 21, we went through some materials that should be familiar from lecture. You should know the 7 characteristics of life, the 4 features that all cells have in common, and you should be able to differentiate between prokaryotic and eukaryotic cells. With eukaryotic cells, we then took a survey through protists, fungi, plants, and animals. The first eukaryote that we looked at was Spirogyra, so you should be able to recognize that this is a protist. Its specific name is Spirogyra. And the cool thing that we can notice here as we look at this are these spiral chloroplasts that we see internally. Note that this is a eukaryote because it contains internal membrane-bound organelles. The next organism that we looked at was a fungus, Rhizopus. So again, you should be able to recognize it's a eukaryote. It's a fungus, specifically Rhizopus. And we saw these structures that again have internal membranous-bound organelles. Here's another picture of that same fungus. The third organism that we looked at was another eukaryote. This specifically was a plant cell called Elodea. Each one of these rectangles that you see is a unique cell. That outer side of the rectangle is where we have the cell wall and the cell membrane, and those little green dots are the chloroplasts where photosynthesis takes place. The last eukaryote that we looked at was an animal cell, specifically a cheek cell. On this, we can see that it's got a cell membrane and it has a nice dark nucleus. Note that because this is an animal cell, it does not contain chloroplast or cells. Next, we moved on to prokaryotes. With prokaryotes, we always want to identify them in 2 ways, by morphology and by stain. So let's practice this in a couple different ways. Morphology is just a way of saying shape. If you see prokaryotes that are spiral shaped, we call that spirilla. If they are lines, we say that that's bacilli. And if they are dots or spheres, we say that those are cocci. Gram staining is a staining procedure that will classify or divide prokaryotes up into one of 2 categories. If they're purple, it's gram positive. If they're pink, it's gram negative. So over here in the corner, this spirilla, we would identify that organism as gram negative spirilla. Here in the blue circle, we would identify those as gram negative bacilli. Or here in this blue circle, that would be gram positive cocci. Additionally, sometimes we add prefixes to our organisms. Staphylo is when we have grape-like clusters. Strepto is when things are in chains. So this top picture, or this purple one, we would just find those prokaryotes as gram positive staphylococci. This bottom picture, we will identify those organisms as gram negative streptobacilli. We then concluded by talking about viruses. You should know what characteristics of life viruses have and what they don't have. Viruses are unique. They're a little bit more than just a chemical or a macromolecule, but they're not quite a cell. They do not contain organization in the fact that they're not made up of cells. Also, they are unable to reproduce on their own. However, they do have a very lifelike quality in that they are able to evolve evolutionary adaptations, as we've seen with coronavirus. So they're this interesting in-between organism that isn't quite a living cell, but it's definitely something more than just a macromolecule. On Unit 3, we explored the scientific methods more. You should be comfortable with the definition of a hypothesis. You should also, again, be able to identify independent variables versus dependent variables. The first thing that we learned in Unit 3 was how to make solutions from stock solutions using this equation, C1V1 equals C2V2. You need to be able to use this equation to answer questions about making solutions from stock. Here's a sample. How much of a 40% stock solution is needed to make a 15 milliliters of a 6% solution? Begin by figuring out what you have. The concentration of our stock solution is 40%. So C1 is 40%. Volume 1, I don't know. That's what I'm trying to figure out. How much of that initial stock do I need? So that'll be our X. The concentration of our new solution that we want to make is a 6% solution. And then we want to make a volume of 15 milliliters. We then plug this equation and solve for x. So 40x equals 6 times 15. This is 90. 6 times 15. 90 divided by 40. X turns out to be 2.25 milliliters of stock. So to answer the question, how much stock is needed, we would need 2.25 milliliters to make that solution. Note that we would still have to add water to dilute that. We would need 12.75 milliliters of water in order to bring up that total volume to 15. Know the Daphnia, know where the heart is. Know that caffeine is a stimulant, ethanol was depressant. Calculations, you should be able to take a given rest and heart rate and calculate a 10% change. Let's do that down here at the bottom. So I have a Daphnia whose resting heart rate is 115 beats per minute. What would a 10% change look like? In order to do this, you take 115, multiply it by 0.10, that's a 10% change, and you get your answer. In this case, the answer is 11.5. 11.5 doesn't answer the question of what is a 10% change. We need to do one more step. We need to go back to what we originally had, which was 115 beats per minute, and then we're going to subtract now that 10% change minus, the 11.5 beats per minute. So if we do this, 115 minus 11.5, the answer would be 103.5 beats per minute. So if I had asked you on a practical, what is a 10% decrease from a resting heart rate of 115 beats per minute, the answer would be 103.5 beats per minute. You should also be able to calculate the average of heart rates at 15 seconds, similar to what we did in the experiment. You should also be able to take that average heart rate and turn it into a heart rate per minute. Once we generated data, you ended up with a graph that looked like this. This is slightly different. We're looking at acetylcholine here, but you should be able to analyze these graphs. So let's see how we might ask you to do that. Let's imagine on a practical, I asked you, what concentration of acetylcholine will give you a heart rate of 95 beats per minute? To do this, you need to find 95 beats per minute. So there it is. You are then going to take a line straight across until you hit your standard curve line and then drop straight down. In this case, we cross the x-axis at 1.2, 1.4, 1.6, at 1.6% acetylcholine. So to answer the question, what concentration of acetylcholine will give me a heart rate of 95 beats per minute? The answer would be 1.6. Hi, Unit 4. So in Unit 4, you should know that proteins are a large macromolecule composed of amino acid monomers. Proteins have a variety of functions because of their primary, secondary, tertiary, and sometimes quaternary structure, which makes them a very versatile molecule. We are specifically looking at a protein called amylase. Amylase is a protein that acts as an enzyme, meaning that it speeds up reactions. In Unit 4, we gave you different concentrations of solutions that had amylase in them, and you had to figure out how much amylase was in those solutions. Know the difference between solutions, solvent, and solutes. Solutes are the chunks or the things that were dissolved. Solvent is the liquid, and the solution is the entire mixture. To figure out the concentration of protein in various solutions, we used a spectrophotometer. The spectrophotometer is a machine which shines light onto a substance and then is able to quantify what happens to that light. You need to know how light works. If we think about our red apple over here on the side, the apple appears red because it's absorbing all wavelengths of light except for red, but then it's transmitting the red back into our light. into our eyes. From that example, hopefully you saw something that absorbance and transmission are inversely related. If absorbance is very high, transmission is going to be very low. So this thing is absorbing all of the red light, but it's not transmitting any of the red light. We can use the spectrophotometer when we add a Bradford reagent to a solution that contains protein. Bradford reagent will change colors when it's in the presence of protein. When there's not very much protein, it stays brownish. When there's lots of protein, it turns bluish. So what we can do is we can add Bradford and then put these samples into the spectrophotometer and read their absorbances. Over here you can see this happening. Here, this brown tube, we put it in. The absorbance was very low. That means there's not very much protein, versus tube B, where the absorbance was really high. It means there's lots of protein. One thing to remember about the Bradford is that absorbance values of greater than 1 are unreliable and should not be used. Before we could figure out the absorbance and the concentration of our unknown solutions, we had to create a few tubes where we did know specific things about it. One type of tube that we created was called the blank tube. The blank tube is going to contain everything except for what you're interested in looking at. So in this case, it had glass, right? We had our test tube, that's going to absorb light. It had water, that's going to absorb light. And it also had Bradford reagent, that's also going to absorb light. What we did was we put that blank tube in the spectrophotometer to zero out the machine or to calibrate the machine. We were telling the machine what it should be ignoring. Another thing that we had to do was we had to create a serial dilution. A serial dilution is where you start off with a known amount of protein and then you dilute it. In our particular case, we diluted it in half each time. So with our serial dilution, we had something where we started off with maybe 100 milligrams per milliliter protein in the first tube, and then we diluted it in half in each subsequent tube. So we knew the next tube had 50 milligrams per milliliter protein, and the next one had 25 milligrams per milliliter protein in it, and it just diluted in half each way. Once we had the standard curve, we could take these tubes where we knew exactly how much protein was in each tube, and we could read their absorbances. This allowed us to create a graph that looks something like this. Protein concentration are independent variable on the x-axis, absorbance are dependent variable on the y-axis. Using this standard curve, we could then finally try to start looking at our unknown solutions. We could take our unknown solutions and figure out their absorbances. So let's try one. Here's an unknown whose absorbance is 0.5. So I took an unknown, I plugged it into the spectrophotometer, and I figured out that its absorbance was 0.5. From here, we can figure out what the protein concentration is in that particular tube. We begin by finding 0.5 on the x-axis. Once we found 0.5 on the y-axis, once we find 0.5 on the y-axis, we then draw a straight line over to our standard curve. Once it hits our standard curve, we then draw a line straight down. Where it crosses the standard curve tells us approximately how much protein is in our unknown. So if you had an unknown with an absorbance of 0.5, its protein concentration is about 13 mg per ml. This is an okay method to use, however, we're just eyeballing. A better way to do this type of experiment is to have a computer and to use algebra and to figure out the actual equation of a line. And it ends up looking something like this. Equations of a line always will equal y equals mx plus b. What will happen is you will know y, you will know that absorbance value, that y value, and then you're going to solve for x. So maybe here's a sample question. I give you an equation of a line, and then I ask, what is the concentration of an unknown protein solution with an absorbance of 0.463? What you would do is you would plug 0.463 in for y. So 0.463 equals 0.39x minus 0.45. You then have to solve for x. So let me turn on my calculator, 0.463 plus 0.45 equals 0.913 equals 0.39x. You then solve for x, so 0.913 divided by 0.39 X ends up being 2.34. Again, this is a better way to figure out the protein concentration because it involves actual math and we're being very precise instead of just eyeballing. You should definitely know and be comfortable with how to figure out the protein concentration of an unknown solution using both the hand-drawn or the box method and the equation of a line. Unit 5, looking at amylase activity. You should be familiar with the reaction that amylase is actually catalyzing. So starch, again, is a polysaccharide. This is a big complex carbohydrate. This is the molecule that we start off with. Amylase is our enzyme. That's our protein. And what amylase can do is it can take starch and it can help it break down into molecules of maltose. Maltose is then broken down further by an enzyme called maltase into molecules of glucose. This is an important reaction because this is a key component of energy processing. Organisms need to create this glucose so that they can run cellular respiration. Enzymes, of which amylase is one, are catalysts. They lower the activation energy. Enzymes have a specific spot on them called the active site. The active site is where the substrate binds. In our particular experiment, starch is the substrate, amylase is the enzyme. When starch and amylase binds, that lowers the activation energy, and then our reaction proceeds, and we're able to make maltose as a product. One of the things that we wanted to look at in Unit 5 was whether our enzyme amylase was specific for starch and cellulose. We know that amylase likes to react with starch. What we're wondering is, does amylase also react with cellulose? Cellulose and starch are very similar in structure. They're both made up of glucose monomers. The difference is the linkage between them, whether they have the alpha or the beta linkages. For amylase sources, we had two different sources. Human saliva was our experimental condition. So when you spit into the cup, the reason we were doing that is we were trying to collect up our saliva to find out if humans in their saliva contained amylase any amylase enzyme. Again, this would be an important enzyme to have because it would help us start breaking down starches. We also used amylase from a commercial source where we bought the amylase from a company. This was a control. I knew it was guaranteed that the commercial amylase would always give you results. Some of the experiments we ran in experiment Unit 5 were qualitative, meaning they were just colors. Others were quantitative, meaning they were numerical. And you should know that starch is the substrate, amylase is the enzyme, and maltose is the product. What we wanted to do in Unit 5 is we wanted to see if our amylase was active as an enzyme. There were two ways to detect enzyme activity. We could, number one, look for a decrease in substrate. If there was less starch, that meant our enzyme must be doing things. Or we could look for, number two, an increase in product. If there was more and more maltose, that also meant that our amylase was active. To do the first thing, to look for a decrease in substrate, we used iodine. Iodine in the presence of starch turns black. Iodine, when there's not starch, turns yellow. So for us, black would mean that the starch is still there. The amylase is not doing anything. If we saw yellow, though, that was a good sign. That meant the starch was being broken apart and that the enzyme was active. The way we did this was with a qualitative experiment looking at the starch plate. We took up starch plate and then we added iodine. Places where it stayed black showed that the enzyme wasn't working. Places that stayed yellow showed that the enzyme was working. What most people saw was that the commercial amylase was very active. It created a lot of clearing. You got a nice yellow pattern. The saliva, our experimental group, also created some yellowing, showing us that yes, our saliva did have amylase activity. Again, here are the colors that you should know. Iodine in the presence of starch stays black. Iodine, when there is not starch, turns sort of a yellowish amber color. A second way that we could look at amylase activity was by looking at DNS reagent. DNS reagent is a reagent that turns sort of a brown color or a dark orange color in the presence of reducing sugars. Maltose is a reducing sugar. So this was a way to look for an increase in maltose. If you got a lot of maltose, if you got a lot of brown color, it meant that your enzyme was working really well. In this experiment, we set up a standard curve for you. This is what the standard curve may have looked like. Again, things with very low concentrations of sugar would be sort of that yellowish color. Things with really high concentrations of sugar would be that darker color. You then set up a series of different conditions. These do not correspond to the letters that they were in the lab manual. I apologize for that, but let's just go through them. In one condition, you set up starch plus amylase. The reason to put starch plus the amylase that we had bought from the company was that this was going to be a positive control for sugar production. I know that the amylase we gave you was good. And so if you take amylase enzyme plus starch product, I know for sure you should have gotten sugar production in that tube. The second tube that you got was starch plus maltose. We didn't even add enzyme. Or in other words, I just gave you a tube where there was lots of sugar floating around. This was a positive control as well, but this was a positive control for the DNS reagent. I knew for sure that this tube had tons and tons of sugar in it, and so it was a way for me to make sure that your DNS reagent was working appropriately. The starch plus the saliva tube, this was our experimental condition. This was the experiment where we were trying to see what would happen. And then in your final condition, you had starch plus amylase reaction buffer. This was a negative control for sugar production. This was a place where I knew that no sugar should be produced. With these, you then went ahead and first you ran your standard curve that we provided. So you got a bunch of values that you could then draw the line through and create. And then you figured out the absorbances of each of these experimental tubes, and you were able to figure out how much sugar was produced in each tube. What you found, not very surprisingly, or maybe surprisingly, was that yes, you got sugar in tube B, but we knew that was going to happen. Yes, you got sugar produced in tube A, but again, we knew that was going to happen. And then interestingly, we also got sugar in the starch plus saliva condition, showing that our human saliva does have amylase activity. We did not see activity in the starch plus amylase reaction buffer, but again, that's what we expected to see. The last experiment, we looked back again at starch and cellulose to see if there was any specificity. So we took our enzyme amylase and we added it and tried to see if we could get any sort of maltose production. When you took the enzyme plus starch, as predicted, you got a nice dark reaction. They worked really well. On the other hand, when you took your enzyme plus cellulose, you got a really duddy reaction. Nothing happened. So what this experiment showed us is that our enzyme amylase is specific for starch. It does not like to break down the cellulose, and it's able to tell those differences. Unit 6. In Unit 6, we continued working with the same reaction from Unit 5. We had starch, which is our polysaccharide or our substrate or our carbohydrate. We then added amylase. Amylase is the enzyme, it's the protein. And we were looking at our product, which is maltose. Specifically, the reaction that we looked at, we used, wanted to look at whether or not starch was there or not. So we did an iodine test. Just as a reminder, if the reaction turns black, that means that the amylase is not working. Iodine plus starch means black, so if there's lots of starch, your enzyme isn't working. If, however, your reaction turns yellow, that means that the starch is disappearing, or in other words, amylase activity is functioning. We wanted to, in Unit 6, figure out different factors that might influence enzyme activity. Some of those factors include temperature and pH. Proteins, enzymes, can denature when they get into suboptimal conditions. So at high temperatures, they may denature. At extreme pHs, they may also denature or lose their native conformation. In the first experiment, you set up a variety of different temperatures. These are different than the ones you ran, but these are just a sample. And then you looked at how the enzyme acted over time at that particular condition. So in this bottom row at 55 degrees Celsius, you can see the enzyme started off and it wasn't working. That's what black means. And then it just stayed black the entire time. So at 55 degrees Celsius, it looks to me like this enzyme is not very optimized. It's not working well. It's probably been denatured. If, however, we look at what's happening at 35 degrees Celsius, you can see that that reaction quickly turns yellow and stays yellow. So what that's showing me is at 35 degrees Celsius, this particular enzyme is working very efficiently. You should be able to take any type of data similar to this type of image and be able to tell me the optimal temperature or the optimal pH for whatever enzyme you're looking at. Again, the thing to remember is black means the enzyme isn't working, yellow means the enzyme is working. With acids and bases, you should know that acids are going to increase hydrogen ion concentration, whereas bases decrease the hydrogen ion concentration. Buffering capacity is how much a solution can resist change in pH when acid or base is added. Buffers are usually a weak acid, weak base pair. You should be able to evaluate the pH using pH paper. So if you take the piece of pH paper and dip it into a substance, you just compare the coloration and then figure out the pH. In this experiment, you did the following. You set up your amylase reaction and you added it to something that contained acid or something that contained base. In this experiment, when you had your enzyme trying to work in the presence of acid or base, it didn't work very well. You then took the same enzyme experiment and you added a buffer with your acid or a buffer with your base. And what we saw when you had the buffer there, those enzymes were able to remain functional for longer, and you did see the enzyme continuing to function. In the last experiment, you looked at cofactors. A couple of things to note. EGTA is not a cofactor. Things like magnesium and calcium, those are cofactors. Cofactors are small, usually inorganic molecules that are enzyme helpers. Some enzymes require them, others do not. What EGTA does is it blocks the cofactor. So let's start with the top picture. Here we've got our enzyme, so think amylase. Here we've got our substrate, so think starch. And then that little black dot right there, that's our cofactor. If it is the case that cofactors are required, you can see how that cofactor helps the starch and the amylase bind together so that we can get our products. What EGTA does is it's a chelator. What it does is it binds to cofactors and makes them unavailable. And so what happens is that enzymes and substrates are unable to bind when the chelator is not functional. In the final experiment, what you looked at was you tried to decide if our particular enzyme amylase requires cofactors to function. Here's what you may have seen. Again, this is slightly different than what you ran. Here's the enzyme running at normal, and you can see, oh, it goes yellow. It's working really great. And then here's what's happening to the enzyme when we start adding EGTA. You can really see at those higher concentrations especially that it just stops working. What do these results indicate? These results indicate that amylase does require a cofactor to function. Unit 7. This material comes from the lab bench exercise that you're doing in Pearson and the associated PowerPoint PDF file that you have to refer to. First thing you should be able to calculate is the surface area to volume ratio of a cube. This is very similar to what you did in the lab bench exercise. To figure out the surface area to volume ratio of a cube, you would do the following. To figure out the surface area, it's length times width times the number of sides of a cube, which is 6. To figure out the volume of a cube, it's length times width times height. So you should be comfortable with those calculations. You should also understand why there are size limits to cells. Why do prokaryotes have to stay small? Why can eukaryotes get so large? But why is there even a limit to how large those eukaryotic cells can grow. We then ran an experiment where you looked at different cubes representing different sizes of cells so you could see how things enter or leave a cell. It's really important to have enough surface area so that things can get into and out of a cell. When a cell gets too large, it's harder for things to get in and out. So you should know the following. We used phenolphthalein and we made some cubes with sodium hydroxide. And so this is where you got those bright pink cubes. So you should know that if you make these agar cubes with base and phenolphthalein in them, they'll turn bright pink. You then took these bright pink tubes and you dumped them into a solution of vinegar. Vinegar is acidic. When vinegar is able to get into the cube, it will change that phenolphthalein from pink to white. So what you got was then these cubes where you could see how far that vinegar had been able to move into the cells. And you should understand, why is it pink? Why is it white? What's moving? Again, it's that vinegar moving into the cell causing that white. And notice that no matter the size of the cell, the vinegar is still only able to move the same amount, whether it's a big or a small cell. Next, we began looking at osmosis. You should know the definition of osmosis. It's the movement of water molecules from an area of low solute concentration to an area of high solute concentration. You should be able to predict this movement in any type of scenario. We use dialysis tubing. Dialysis tubing is a tubing that allows water to transport, but not another molecule such as sucrose. So again, if we think about this particular situation, water is going to move, because sucrose can't move in this scenario, from an area of low solute concentration to an area of high solute concentration. Or in other words, water is going to be moving out of this baggie into the beaker. With this, we want to use the words tonicity to talk about the solutions. Tonicity is always a comparison. When you are comparing two things, if they are the same solute concentration, those are called isotonic. If something has more solute in it than what you're comparing to it, we could describe it as hypertonic. If it has less, it is considered hypotonic. In this particular situation, the 5% sucrose solution is considered hypertonic in comparison to the 1% solution. On the PowerPoint notes, we then looked at diffusion. Diffusion can be influenced by the size of the molecule and the temperature. The size of the molecule, the larger the molecule, the harder it is for it to diffuse because it's so big it can't really fit. The smaller the molecule, the faster it can diffuse. So in these two circles, if I'm looking at them, I know that the blue molecule is a smaller molecule because it diffused faster. The red molecule is a larger molecule because it diffused slower. Also, temperature can affect diffusion rates. Hot things have more motion, so hot things are going to diffuse faster than cold things that are slower and don't have as much motion. Next in the PowerPoint, they set up the following reaction. They took dialysis tubing and filled it with starch, sodium chloride, glucose, protein, and they also mentioned filling it up with sulfate ions as well. They then allowed this substance to sit there for some time, and then they checked after time had passed to see if any of those 5 substances were able to exit the bag. For each of these molecules, you should know the test that we used and the colors they turned. For starch, we used an iodine test which will turn black. For sodium chloride, we used a test called silver nitrate, which turns white if it's positive. For glucose, we did a test called the Benedict's test, which turns orange if it's positive. For protein, we did a biuret test, which turns purple if it's positive. For the sulfates, we used a test called barium chloride, which turns white if it's positive. Be able to match each of those substances up with their tests, also their colors. After running this experiment, what we found is that only some of the substances were able to make it out of the bag. The sulfate ions were able to make it out of the bag. The chloride ions were able to make it out. The other items were too large, and so they remained trapped inside the dialysis tubing. This was an experiment looking at diffusion because we were looking at molecules moving, not water. From there, we then looked at what happens to animal cells and plant cells in different tonicities. You should be familiar with what happens to animal cells placed in different tonicities, as well as plant cells. Please make sure that you can use the single term that's used to describe each. So for instance, if I ask you, what happens to an animal cell in a hypertonic solution, you would respond that it becomes shriveled. Or what happens to a plant cell in a hypotonic environment, it becomes turgid. You looked at images first of animal cells by looking at blood cells. If you put an animal cell into an isotonic environment, they remain normal and they're nice and round. If you put an animal blood cell into a hypertonic environment, they don't become darker. That's just the lighting of the microscope in this particular image. What happens is the edges of the cells become irregular. So they're not really getting that much smaller. They're just getting sort of weird around the edges. So isotonic is nice and round. Hypertonic is sort of funny shaped. The isotonic ones we describe as normal. The hypertonic ones we describe as shriveled or crenate. If you try to put animal cells into a hypotonic environment, they will immediately fill up with water and they will continue expanding until they explode. So there isn't really an image we can show you for that. With plant cells, if you put plant cells into a hypotonic environment, the vacuole swells up and this pushes the chloroplasts out to the edges of the cell. So you see those chloroplasts mainly focused on the outside edges of the cell. These cells are called turgid and they're very happy at this condition. If you put plant cells into a hypertonic environment, they actively lose water from their vacuole. The cell remains the same shape, but now what's happened is most of those chloroplasts are on the interior portion of the cell. This is called plasmolized. This is not a great option. The cell is dying when this happens, but you can see the difference there by focusing on the chloroplasts. That gets us to the end of this review. Again, I went really quickly. I didn't go back and edit. I may have misspoken. If you have any questions, please definitely reach out to your instructors, and good luck studying for the practical.